Sea stars attach to surfaces with an unbreakable grip underwater. In this video, we reveal the research behind unlocking their secret adhesive mechanisms. The problem is that synthetic adhesives struggle to bond in wet environments like water or blood because water disrupts the bonding process, leading to poor adhesion. Frustrated by this, a team of 12 scientists turned to nature for inspiration. After long hours at the lab, they published their breakthrough paper. Sea star-inspired recombinant adhesive proteins self-assemble and adsorb on surfaces in aqueous environments to form cytocompatible coatings. Imagine having underwater repair materials that never fail or surgical glues that effortlessly seal wounds. After studying sea stars, researchers discovered a special adhesive protein called SFP1. Since SFP1 is enormous, about 3,800 amino acids long, they focused on synthesizing the key sections responsible for binding. To do this, they cloned, expressed, and purified these recombinant protein fragments using E. coli, a common bacterial workhorse. However, the process wasn't straightforward, as they had to overcome challenges like insolubility and aggregation, where the protein tends to clump together. The exciting part was when they used circular dichroism and atomic force microscopy, techniques that act like a molecular MRI, to verify the protein's structure and binding performance. And the results were interesting. They successfully synthesized two fragments of the sea star adhesive protein, SFP1. The recombinant SFP1 beta C term achieved 95% purity, while the SFP1 delta fragment reached 75% purity. The researchers then exposed these protein fragments to various salt concentrations, mimicking seawater conditions, to observe their behavior. Excitingly, both fragments self-assembled into nanostructured coatings, although each fragment behaved differently. One formed dense meshworks with structures ranging from 80 to 250 nanometers in size, depending on the salt type, while the other only adsorbed onto surfaces in the presence of calcium ions, forming structures with heights between 600 and 800 nanometers. Remarkably, these nanostructures mimic the natural adhesive footprints left by sea stars. Finally, when tested with human cells, the coatings proved not only non-toxic, but also promoted cell growth. Now, based on these results, the research opens up a wide range of opportunities. These bio-inspired adhesive coatings could be applied to implant surfaces, potentially reducing the risk of rejection and improving integration with human tissue. Another cool aspect is the ability to self-assemble in wet environments, paving the way for developing advanced surgical adhesives. Now, here's my opinion. Although the results are impressive, it would be interesting to see whether the coating actually improves adhesion underwater, not just its ability to self-assemble. Another cool thing would be to try to synthesize the whole SFP1 protein instead of only two fragments, which could reveal more potential. And now the meme of the week. Thanks for watching, take care, and see you next Wednesday. And if you haven't already, check out my video on how spider silk is reinventing arteries.